It's a great pleasure to introduce Jack Withrow, who is from Kodiak, where his parents still live and work. And Jack got his undergraduate degree at Western Washington University and came to us as a master's student some years ago. If he's a familiar face, it's because he's been around a little bit longer than most master's students, and that's because he got a full-time job in the course of his degree study, and has, when Dan Gibson retired as collections manager of the bird collection over in the museum, Jack Withrow was the successful applicant to be his successor. So Jack is now collections manager for the bird collection in the museum, and has, with a full-time job, his thesis became his hobby for a little while, and we'll see today that that hobby has been completed, and certainly he'll find some other ways to spend his free time now, if he's successful. With that, let me turn it over to you.
Thank you for coming. If you haven't figured it out already, I'm going to talk to you today about speciation or divergence in two pairs of high-latitude birds. So birds are generally thought to exhibit allopatric speciation.
Different font? Great. We'll pause for this
Auspicious
section.
start.
There are many copies of Thesis Talk. That one's dated this morning at 9:05. That's probably a good sign.
Yeah. So, we went through that one already. And because most birds are allopatric, or thought to speciate allopatrically, it's more interesting when funny things are going on. And today I'm going to talk to you about my search for speciation with gene flow. Speciation with gene flow is more likely to happen when things have a parapatric range. And what I mean by that is when they're largely allopatric, but their ranges sort of overlap in the small area of sympatry. We call it parapatry. And heteropatry is just a cyclic period of sympatry throughout an annual cycle. It's very common in migratory birds. So characterization of these processes improves our understanding of speciation with gene flow, which is well understood theoretically, but there's very few good empirical examples of it. So studying the genomics of speciation with gene flow, predominantly in these parapatric and heteropatric scenarios. And there's two main ways to increase your chances of finding evidence for this. One is to study taxa that are young. When you study young taxa, you're more likely to find evidence for the processes that led to their divergence. So if they're close to the species threshold, you're more likely to find evidence for what caused them to cross that threshold. The second way you can maximize this is to study taxa where currently they have a pattern of parapatry or heteropatry, and that this pattern is likely to be the case historically as well. There's lots of things that can inhibit your ability to infer the speciation process. I'll run through a list here. Uncertainty about geographic ranges, past geographic ranges. People have tried to mitigate this with things like niche modeling, but for the most part, geographic ranges of the past are highly speculative. We're also uncertain about initiation and divergence times. These things can be mitigated, but finding concrete answers is hard. And also people tend to discount major phenotypic attributes, things like migration. Things like migration are highly evolutionary conserved, and when you see it in a taxa today, it's almost certainly happening in a taxa in the past, and that migration tends to increase the mobility of organisms and tends to make them disperse more and increase in gene flow, essentially. And so I'm going to talk to you today about two systems that I feel good systems to find speciation with gene flow. We're going to test two very simple hypotheses in that. The first is that we're going to find genomic divergence. And this is, you may think this is overly simple, but one of the systems I'm going to talk to you about today is a subspecies, and most bird subspecies don't show any genetic differentiation. It's rare to find genetic differentiation in a subspecies. And one taxa I'm going to talk to you today about is an Arctic high-latitude species, and they often show shallower divergences than, say, their tropical counterparts. And secondly, if we're looking for gene flow, I'm suspecting that I'm going to find some evidence of this. So we're going to approach these two hypotheses in three broad ways. The first is with a quantification of genetic divergence in population structure. Second, I'm going to estimate divergence times, population sizes, and gene flow. And thirdly, I'm going to assess genetic diversity in selection. Before I get into the dynamics of the first chapter here, I'm going to kind of lay out where I'm going with this. There's two systems. I'm going to run you through the sort of the natural history, the demographics of both populations, and then since the methods are so simple or so similar in each system, I'm going to sort of go through a joint methods. And I've got little plovers down here and owls in previous slides that'll sort of orient you. And that whatever taxon is at the bottom of the page, that's the one I'm talking about. If both of them are down there, I'm talking about both. If it's not apparent from the title. So that's the working title of the first chapter. The reason I wanted to look at these guys is because, as far as we know, they're young. They were elevated to full species status after a long period of recognition as subspecies in 1993. It may seem like a long time, but in birds, where they're so well known, that's a recent elevation. They're also extremely phenotypically similar. We'll get to that in a moment. And they're also, based on the range and their natural history traits, we felt like they were a good example or a good place to look for speciation with gene flow scenario. And the best way to illustrate that is with a range map. And you can see here that the dark purple is an area of sympatry, mostly allopatric ranges, but they do occur together in western Alaska. Fulva here on the left migrates down to the South Pacific, Southeast Asia, Australia. Dominica is more of a pampas bird that goes to the pampas region of South America. They come up in the summer and breed in the Arctic, and some birds breed sympatrically there in the Seward Peninsula. So they're both heteropatric and parapatric. So this is a close-up, obviously, of the Beringian range. What you should take from this is that during the glaciations, the area here would have been much greater. Presumably, it would have been okay habitat for Dominica and Fulva to breed. They were probably still migratory back in the day, and it's at least as likely that they had some contact during the glacial cycles as it is that they were completely geographically separated during that time. These are just some pictures of the study taxon. Dominica on the top, Fulva on the bottom. The modeled birds are females. The two birds in the middle are males. This is the kind of differences that you'd expect to see in a subspecies in a bird. They're very, very similar phenotypically. You need either a bird in hand or point-blank photographs of a male bird to be able to distinguish these taxa. We give you the rundown on the owls. Heteropatric differentiation in Haida-Gwaii owl. Agallis caticus brooksii. Haida-Gwaii is just the native name for the Queen Charlottes off of British Columbia, Canada. Again, these are punitively young taxa. They were only recognized as subspecies at the moment. There's some previous genetic evidence that suggests that there's differentiation, but we can be confident that they're relatively young. Again, based on their ranges and demographic natural history traits, we feel like they're a good case, in this case, of heteropatric speciation. Again, a range map will serve to illustrate that. So they occur across North America. The nominate bird, nominate being Agallis caticus, is migratory. It generally leaves the Seward Peninsula, or the Kenai Peninsula, higher latitudes of its range, and migrates down. This is about the southern extent of its wintering range. But what that results in is a cyclic movement through Queen Charlotte's during migrations in the fall and spring. It moves through Haida-Gwaii. Again, just an example of what these birds look like. This remarkable amount of differentiation for a nocturnal bird. This is the kind of thing where if you had major differences in other life history traits, you'd start to think that maybe these guys were on the verge of speciation. Now we're going to go through molecular techniques. Use mitochondrial gene sequencing of NADH dehydrogenase subunit 2. We use that in both taxa. For the owls, I also use Site B. These are well-known markers in birds that we use all the time. We kind of know how they behave well. They have good models of evolution. The second method was amplified fragment length polymorphisms. For those of you who are not familiar with that, it's basically a fragment-based technique. You take a restriction enzyme, chop up the DNA, put primers on, ligate primers on there, and amplify a subset of those primers, and what you're left with is a banding pattern. Basically, you're left with a binary matrix that you can then deal with. A relatively cheap way to assess genome-wide diversity. You get lots of unlinked markers or supposedly unlinked markers, and they have been used extensively to study shallow divergences in these kinds of organisms. There's a few cons to them. They are a dominant marker. You cannot assess heterozygosity from them. Consequently, there's fewer software packages for their analysis. So this is a condension of a lot of information, but basically those are the markers we use for pluvialis. The N is the sample size. About half of each of those numbers was in one tax, one species versus the other. Similarly for the owls, about 20 individuals of each marker from each subspecies. So using the mitochondrial DNA, we constructed haplotype networks using network. We calculated phi-st and p-values in Harlequin. We also calculated nucleotide diversity and haplotype diversity, both of those in DNASP. For AFLPs, we calculated FST and p-values in AFLP-serve. We also calculated the likely number of clusters using Bayesian clustering algorithm and structure. We counted the number of fixed loci in one population, but that were polymorphic in the other. We also counted the number of loci that were present in one population but absent in the other. And we sought outlier loci in a simulated data set using DF-DIST2. We also used isolational migration modeling to estimate divergence time, T down here, effective population sizes, and gene flow. This is the sort of exact model, if you will, for plovers. For owls, we use a slightly more complicated model. We can go into the differences there, but for the most part, we ran multiple simulations with useful natural history prior, generation times. This is more detailed than you guys need to know, but it's there if you want to talk about it. So this is my segue into results from the plovers. This is sort of a method slide. It's the exact areas that we took birds from. Basically what you need to get out of this is that we took about half of the birds of both species from the Seward Peninsula and about half from areas away from the Seward Peninsula. So what we had then was a sample from a sympatric area and samples from allopatric areas. This is the haplotype network for our plovers, which shows you basically everything that was going on with them. But what you can see here is that there's very few haplotypes for a bird that's distributed essentially across the Polo-Arctic. Compared to other birds, this is very few haplotypes. It's also much, much older than we thought.
Jack, are those little dots around the edge of those singletons?
Those are singletons, yeah. And each, that's one, so one difference from this, it's 20-some individuals. There is one hybrid here. I'll explain this later. Basically had a mismatch with its AFLP data. FST, or phi-st is fairly high, 4.7%, so that's an uncorrected distance. So much older than we had previously thought. So as I said in the sampling design, when we initially had designed this, we felt that these populations were probably not nearly as divergent as they were, and so we sampled, this would have say been the Seward Peninsula and then the non-Seward Peninsula. What we were looking for was a depressed FST in an area of sympatry. And since they were so much older than we thought, this is less interesting than it otherwise would have been, but basically they're the same between sympatric and allopatric populations. Not terribly surprising given the age of the taxa. A lot of numbers here, but this is the results from our structure results. What you need to get out of it is basically that structure thought that the most likely number of clusters, and this is the actual diagram of that, each vertical line here is one individual. It's rather uninteresting, but what you need to take away is that most individuals had like 99.9% of their genomic material originating from one population or the other. You can see here that this is the hybrid. About 99.9% of its genomic material is from Dominica. If you remember from the haplotype network, it had the common fulva ND2 haplotype. And so this suggests to me that it's not an F1, probably not an F2, it's an old speciation event. And given the level of divergence we see in mitochondrial DNA, it's probably not, it's almost certainly not incomplete lineage sorting. So there's some evidence in the plovers for hybridization, although it's extremely rare, and we sort of found it in a roundabout way. Again, a lot of numbers. These are the results from the IM analyses. Basically what you want to key in here is the estimates using a divergence rate of 2.6%. These are mutation rates, so you double it for a divergence rate. So we did calculate them for multiple mutation rates. But I'll key in here on the ones for 2.6% divergence. Effective population size of 16,000 individuals. Migrants per 0.8 million years, which is much longer, older than we had previously thought. These are current estimates for, these are census sizes, I guess, so they include males. This number here only includes females. What you should take away is that it's the same order of magnitude, roughly, as our current population estimate sizes. So these are the actual posterior probability density curves, or the numbers that I just showed you. What you can see is a nice peak here at 1.8 million years. Similarly for migration rate, almost all of the distribution is here at 0. The 95% confidence interval would basically include 0. Again, a nice peak at around 16 or 17,000 individuals. Nucleotide diversity was lower in fulva. The haplotype diversity was similar between the two species. AFLP data, we generated 242 loci, almost 60% of which were polymorphic. There was no significant differences in loci fixed in one population, but polymorphic in the other is roughly 15 in each. And the number of differences, the number of loci present in one but absent in the other, similarly, no significant differences. There's about 10 in each population that were present but absent in the other. This is the results of our simulation looking for selection. You can see here is that 5 loci were denoted as outliers. You'd expect roughly 2.4 to be seen as outliers. But this pattern of this long tail of high FSTs and high heterozygosities is not something you normally see in these. And again, because of the oldness of the divergence of these birds, I'm not really inferring selection here. This is something that's probably a result of the higher divergence than this program is designed to test. So the answers to the hypotheses: yes, there's way more genetic divergence than we had previously thought, 4.7%. FST in AFLPs is about 2.2. So these are not a recent divergence. They are, however, a good example of a cryptic diversity event in the Arctic, something that's much more common in the tropics. And so we did not find very much evidence for gene flow. We did find one hybrid. IM suggests that historical levels of mitochondrial gene flow are very, very low. So basically, in the plovers at least, we did not find evidence of speciation with gene flow. Given that they're as different as they are, there is a high likelihood that they have strong isolating mechanisms. Migration is probably one of the big ones that leaves all sorts of differences in phenology, timing differences, wintering ground adaptations. There is differences in calls, so there's probably some sexual selection. They breed in slightly different habitats, so there's probably some breeding habitat selection, possibly. And the point is that although these mechanisms could probably have occurred in parapatry, they are also as consistent with a divergence in allopatry. So it's a little bit hard to say how these plovers speciated at this point. Again, I'm going to switch now to the results from the owls. This is sort of a method slide, but what you can see is that we sampled most of our birds from the Pacific Northwest. We sampled all the owls that we could get our hands on from Queen Charlotte's, and we included a few widely distributed species sort of to get a handle on continent-wide genetic variation. Haplotype networks. Again, these are fairly few haplotypes for a bird that's distributed across North America. The surprising thing that was the site B here, where there's only 3 haplotypes, but there's a fixed difference. So it's sort of no surprise that the phi-ST values here are significant. The ND2 one sort of drives the combined. But this is a significant level of genetic diversity or divergence in a subspecies. Again, these are the structure results. What you need to take away is that the structure thought that 2 population clusters was the most likely. This is the actual structure diagram. It's a little more interesting than the plovers. The really interesting thing, though, is that even though these guys are very young, they're only subspecies. With their nuclear markers, we were able to distinguish 2 distinct populations here. And I'll come back to and speculate about these admixed individuals in a moment. As I said, we used an IM model that was a little bit more complex than the owls, so I'm going to present the results slightly differently. Divergence time was estimated at 16,000 years. The migration rates, similarly very low. I'll show you the curves in a moment. I didn't have enough room to put the posterior probability distributions on here. Effective population sizes, those are in females. It's basically a long-term effective population size of females. So Catechus here is 2 orders of magnitude larger than Brookside. And S is a splitting parameter. It's basically a measure of what proportion of the ancestral population gave rise to Brookside. There's some evidence there based on this very small s that there was some sort of founder effect, not unexpected given the island distribution of these birds. And these are current best estimates for the number of Brookside and Catechus. These again include males. These are census sizes. What you should take away is that they're the same order of magnitude for both Brookside and Catechus. Catechus being much larger population size than Brookside. These are actual curves for migration rates. They're not quite as slammed up against the left here as the plovers, but most of the estimates are close to 0. That's what you need to take away from this for both directions of migration. Again, you have a clear peak here. This is that around 16,000 years ago. These are effective population sizes. An okay peak for Brookside. Catechus did not converge, as we say, although it did show that there's a very high population size in Catechus. It's possible that this didn't go down to 0 because there's a lack of, possibly because of the lack of information, enough information in the mitochondrial DNA in this model. So nucleotide diversity was lower in Brookside, even though haplotype diversity was lower, it was not significantly so. For the AFLPs, we generated 405 loci. Much fewer of these were polymorphic, only 25% compared to the plovers. There was, again, no significant differences in the loci, number of loci that were fixed in one population, but polymorphic in the other. And there was no significant differences in the number of loci present in one population, but absent in the other. This is our output from DF-DIS, again looking for selection. There was fewer loci here, 1, but this is not, you would expect at a 99% confidence quantile, C4, so basically what we see from it, see from this is that breast cancer. We don't see evidence of selection here, and even though, I'll leave it at that. We're not seeing an evidence of selection here. We know that these owls have differences in migration, differences in plumage, differences in dietary habits, so it's almost certain that they experience some selection. It's just not, we're not seeing it in the loci that we sampled here. And so basically, coming back to the hypotheses, there is a significant amount of genetic differentiation in the owls, especially given their current status as mere subspecies of one another. And then gene flow, it's effectively we found very little evidence for gene flow in females. And the AFLPs, as you saw, they broke them apart very well. It suggests that a significant period of assorted mating has occurred. And then, if you remember the structure diagram where there was a few individuals that were admixed, so to speak, they showed a structure that suggested that some of their genetic material come from the opposite population. Given that they're, one, a very young species, and two, there's almost no female-mediated gene flow, I suggest, I guess, that that admixture is a result of incomplete lineage sorting in a young taxa and not evidence of ongoing gene flow. This is sort of bolstered by the fact that owls, like most birds, have female bias dispersal. That means the females move the most, and particularly in highly migratory bird like Boleus acaticus, that may even be nomadic. There's a lot of movement. If you're going to see evidence of gene flow, you probably see it in the mitochondrial markers. So in summing here, we did not find concrete evidence in the genetic data that these owls are experiencing heteropatchic speciation, but they are experiencing some strong isolating mechanisms. In the owls, it's probably due to migration habits and dietary differences. However, our genetic data is consistent with a process of heteropatchic divergence or allopatchic divergence, but given geographic and demographic factors, we feel like it fits a pattern of heteropatchic speciation well. General conclusions, we found strong genetic divergence in both systems. There's evidence for gene flow in the plovers, actually, which are much more divergent than the owls. Nuclear diversity and haplodiversity are both low in these groups, lower than a comparison to other groups would suggest. The plover, basically the take-home message is the plovers are too old to assess how they speciated, and the owls have some mitigating evidence that suggests that they experience heteropatchic speciation. And after thanking these people, I'll take any questions.
Yeah, so with the admixed owls, was there any sort of geographic distribution to those individuals that might be insightful?
Those birds were all from southeast Alaska, but that's also where most of our samples were from.
Okay.
And the ones that were the few that were admixed and they were actually Brookside looked like Brookside. They weren't paler or anything like that.
In the plovers, their current distribution has them being seasonally parapathic, essentially, right? Has anyone modeled their distribution in the past to see if that might be a relatively recent phenomenon? Which would be consistent with your estimates of divergence time?
Not that I know of. Like niche modeling, you mean?
Yeah, just to see if this might be, again, a relatively recent phenomenon, that they were perhaps truly allopatric during their divergence.
I think that's most people's working hypothesis, that they were allopatric, but given that their area of sympatry right now is Beringia, and Beringia was much larger in the past, it's at least as consistent that with that, with the dynamic that we see, that it's that was not the alopecia doesn't necessarily have to be the historic.
Then I had a question about the owls. Is the Haida Gwaii owl a conservation concern?
Sure, anything that's only, that they census is less than 2,000 individuals, I would suggest is.
But officially, is it recognized by
Yeah,
British Columbia as-
COSWEC or whatever the Canadian equivalent of our Environmental Protection Agency, I think, would give it. It's on people's radar if that answers the question. No.
So it's related to Mark's question, but so you said that those are other mixed ones that from South East Alaska, right?
Yeah.
Are they captured in the breeding ground or that captured during the winter time?
Most of those birds were salvaged, which means they have a year on them,
Uh-huh.
and so they don't necessarily have a date.
Oh,
And I,
okay.
off the top of my head, I don't know what date is on those birds.
Can you remind us about how like their pairing occurs in like a
Usually the male has a territory and they sing, and then I'm assuming that would attract the female then. So there is some I guess sexual selection probably for song, but why these nominate birds don't stop and breed is something I don't necessarily know the perfect answer to.
So basically, those bonds happens in the breeding period. So the wintering ground doesn't matter much.
Yes.
Okay. Then one more thing, maybe I will forget, so I will ask now. But in the selection plots.
Yeah.
For the clover. Can you bring it up? Why had not the clover, that owl, right? Why are there a lot of low size heterozygous higher than 0.5?
I'm pretty sure it has to do with the way that DF-DIS calculates them. It's not a strict heterozygosity. It's more of a. It's not a strict heterozygosity. Or just leave it at.
So it's not misidentified parallel kind of things.
No, I don't think so. I think it's this long tail is unlike most of these DF-DIS plots that I've seen. Sort of they peter out about here, and this long tail that keeps going. Where of both high FSTs and high heterozygosities is probably an effect that's happening because these things are so different. There's so many fixed differences between them. They probably don't meet the criteria for the model.
Oh,
I mean,
okay.
it's an unexpected pattern about that.
So the heterozygosity in the x-axis is similar to FIP. Total heterozygosity in the entire data set.
I think that heterozygosity is actually has to do with pairwise comparisons within an individual, but I'm not certain of that.
Oh, okay.
Chris?
Your mitochondrial network for the plovers indicates rapid expansion very recently, which seems odd considering that at least for the Eurasian form, it existed or it exists now in areas that weren't glaciated at the time. So do you see any evidence or have any hypotheses as to why you would see this sudden star-like phylogeny, which indicates a sudden expansion rather than something that's got some more time depth to it?
Not really. There's been some, I would call it speculation that market hunting depressed the Domenico's population. No, that's not the one you're asking about, but why fulva would show that sort of pattern, I'm not sure.